We're going to take a look at culture media and obtaining pure cultures. So culture media can either be solid or liquid, and it contains all the nutrients that's required by the microorganism to grow. And we can classify the culture media in a couple of different ways. So depending on whether it's solid or liquid, and then also we can classify it based on its chemical composition. So whether it's chemically defined culture media or complex culture media. And then we can also define it based on its function as well. Let's take a look at the physical nature of the culture media. So it can exist in either a solid or liquid form. If you want a solid culture media, you're going to add agar to it. And agar is a polysaccharide that's made from red algae, one of the eukaryotic microorganisms. And that's really good for obtaining pure cultures. So when you obtain a pure culture, you need to be able to spread the microorganisms out so you can get these individual colonies like we're seeing on this petri dish here. Because then you can pick them off and then go identify them and find out which species that is growing there. And so the microorganisms can't eat the agar, so that makes it really nice to obtain pure cultures by using this solid culture media. You can have certainly put the solid culture media in different forms. You can put it in a petri dish. You can also put it into a tube. And then if you don't want solid culture media, you could also just not add the agar to it, and you could just have liquid broth instead. And so all of the culture media is going to have all the nutrients and chemicals that that particular microorganism is going to need to grow. The next way to classify culture media is based on chemical composition. So chemically defined culture media just means that all of the components, you know exactly what's in there and the exact amount. So you can see in this particular chemically defined media, you can see all the chemicals there, you know exactly what it is, and you can see the exact amounts there. And then compare that to complex media. And in complex media, there are some components there that you're not exactly sure what's in it, or maybe the exact amount may not be known. So for example, in this complex media, we have beef extract. So we're not exactly sure what chemicals are in beef extract. There could be a number of things that are in beef extract. So that's one thing that's making this complex media. And then also the peptone, the partially digested protein, that's another ingredient that would make this complex media, because we're not exactly sure what kind of proteins are in peptone either. And then the last way to classify culture media is the functional type. So we'll take a look at reducing, selective, differential, and enrichment culture media. Okay, so reducing culture media. This is really important to remember. This is going to be used for anaerobic cultures. So if you're trying to culture obligate anaerobes, and remember that obligate anaerobes, they are killed by oxygen. So you can't have any oxygen around because that's going to kill them. So the reducing media actually has chemicals in it that is going to bind to any oxygen around and take it out, so it's not going to be there to kill the microorganism. And then we're also going to put the petri dishes into a container that we'll put a lid on it so oxygen can't get in, and then we usually put in a little gas pack, they call it, that has some chemicals that when you rip that open, they get released, and then they will combine with any oxygen that's left in there, and they will also get rid of the oxygen. So it creates an oxygen-free environment for those obligate anaerobes to actually survive and grow, so you can culture them. And we also have these anaerobic chambers, which are a little bit more fancy. So What they do with this is they have a vacuum system here where they will suck out all of the room air and replace it with mostly nitrogen gas. So there won't be any oxygen in here at all. And you can place your petri dishes with your obligate anaerobes that are growing in there. And so that will keep out all the oxygen, and then you can grow those obligate anaerobes. Okay, selective media, if you look at the name, it tells you what it's going to do. So selective media is going to select for the growth of some microorganisms and then inhibit the growth of others. So a great example of selective media is called McConkey agar. So McConkey agar selects for gram-negative bacteria, and it doesn't let gram-positive bacteria grow on it. So if you have bacteria growing on McConkey agar, then you know that it's got to be gram-negative bacteria because it has chemicals in it that inhibit the growth of gram-positive bacteria. Okay, so selective media selects for the growth of one type of bacteria and inhibits the growth of others. Differential media, the name says what it does. So if you have bacteria growing on differential media, it's able to differentiate between different groups of microorganisms. So for example, blood agar. This type of culture media, they actually add red blood cells to it, so that's why they call it blood agar. And remember we talked about streptococcus and the different hemolytic groups. So the blood agar is actually differential media. So it lets the streptococcus bacteria grow, but it's going to differentiate between the different types of streptococcus bacteria. So remember we talked about the alpha hemolytic, beta hemolytic, and then the non-hemolytic strep. And then another type of differential media would be the McConkey agar. So not only is it selective media, but it's also differential as well. So it's selective in that it selects for gram-negative bacteria. So the bacteria growing here on the McConkey agar is all gram-negative, but it's also differential in that it's going to differentiate between these two different types of gram-negative bacteria that are growing here, and those that are able to ferment lactose, so they're able to break down lactose as a food source, they will actually turn pink. So when they break the lactose down by fermentation, they produce some acid, changes the pH, and there's an indicator that will turn red. And if the gram-negative bacteria aren't able to ferment the lactose, then they don't change color. Okay, so that would be a differential media. They differentiate between different groups of microorganisms. Okay, and the last type of functional culture media is called enrichment media. So this is where you've got a particular microorganism that you want to favor the growth. So you're going to add its favorite food to it, okay, to the culture media. And that'll allow it to grow in larger numbers. So then you can have a bigger sample of it to isolate. So for example, maybe you want to grow more of bacteria that are able to use oil as a food source. So you could actually add crude oil to the agar here as the food source, and that would favor the growth of those bacteria that are able to break down crude oil as a food source. So that would be an example of an enrichment media. And then here's just another example of an enrichment media. We've got a sample here. We've got a number of different bacterial species here, but we want to favor the growth of these red bacteria. So we're going to add the favorite food for the red bacteria. So we add that favorite food, and you can see the red bacteria are going to grow more than the other species, and that allows us to isolate out that red bacteria. So that's an enrichment media. And this is just a nice table that summarizes some of these vocabulary words that we just went over and gives you the definitions there. And the last thing to talk about is isolating pure cultures. This is really important in medicine. So when you send in a sample from a patient, maybe they have a UTI, and sometimes you might have more than one bacterial species causing the UTI. And you want to be able to get a pure culture that just has one species in it so you can identify, hey, what bacteria is this? And then you also want to be able to test that bacterial species to see what antibiotics are going to work to kill it. Okay, so that's why we want to get these pure cultures. How we get these pure cultures, we're going to use the streak plate method. Okay, and we're going to just show you this in a picture, and we'll talk about these colonies on the picture. Okay, so in lab, you would be doing this. What you would do is you would take sample from the patient's urine. You'd have a little inoculating loop, and you would start here at the top of your plate, and you would just streak that across the top of the plate with your inoculating loop, and then you would stick your inoculating loop into a flame, and that would burn off any bacteria that were remaining on the inoculating loop because you want to sterilize it. And then you would take your inoculating loop and start over here on number 2, and then you're just going to take a few of these cells from the first streak and then streak them over here to separate them out even further. And then you'd flame your loop again, sterilize it, get rid of any bacteria that were there. And then you take your inoculating loop and do your third streak, and then spread some of those cells out. And this is what you would get after incubating your plate for 24 hours. So you can see that your first streak, the bacteria are growing really close together, so it would be hard to separate them out. And so if you have 2 or more species, they would all be mixed together, and so it'd be hard to get a pure culture there. And then here's your second streak. They're still kind of all mixed up there. But now on your third streak, you can see some of these individual colonies here where they're separated out. Okay, and these colonies have started either from a single bacterial cell or maybe just a few. Okay, so that's what's going to start these colonies. And then they divide over the 24 hours that they've been incubated, and then you can finally see them. And you can see that there's more than one species here of bacteria. So you can see that some of these colonies are red, and then some of these colonies are more of a pale color. And so you can actually then take an inoculating loop and pick off one of these red colonies, and then you can go and do further tests and identify what that is, and do tests on that to see what species that is, what antibiotics will work to kill that. And then you can also pick off one of these tan colonies and do tests to identify that species and what antibiotic is going to work to kill that. Because maybe this patient in their UTI, maybe their UTI is caused by E. coli and Proteus. And so we'd want to know that, hey, there's 2 bacterial species here causing the infection, and we want to know what antibiotic is going to kill each of these bacteria. Okay, so that's why we would want to have these pure cultures.